This is for a lab you can do right in your kitchen. We're gonna extract some DNA. Here's some things you might need. You need a half cup measure, you'll need a spoon, you'll want some regular dish soap, you'll want some isopropyl alcohol, rubbing alcohol, some salt, a glass with a little bit of water, probably a cup of water in the glass. Start by adding water to the glass and a spoonful of salt. Stir the salt and make sure it's dissolved. What we're going to do next is swirl this water for a full minute. So it's salt water, you got to be careful you don't actually drink it. So that's one safety concern. And a minute is a long time. So you have to brace yourself to deal with pretty rotten taste for a while. All right, so here it goes. Finally, so I spit the salt water back into my glass here. Now, rinsing with that salt water is going to get some cells from my cheeks and mouth. Now, those cells have a membrane. The membrane is made of a lipid bilayer. So to help break that lipid bilayer apart, I'm going to add just a little tiny bit of dish soap. And I'm going to start to stir the water with the dish soap. Now, I'm going to want to stir for a while here, but I want to be careful not to make a lot of bubbles. So I'm not trying to churn the water up in order to make it really foamy or frothy, but I am going to stir it for a while. What this does is helps to separate some of those cells. By separating some of those cells, we can start to break the cell membranes apart, and then the nucleus, we can break it apart, and that'll release some of the DNA. And that's what we're really after. We want good results with releasing the DNA. So I'm going to stir for a while. One of the techniques I found is the longer you stir, the better your results. So kind of keep that in mind as you're stirring. And if it does get bubbly, just get a paper towel and just barely dip the paper towel into the bubbles, and the paper towel will help pick up some of those bubbles and keep your water from getting too foamy from the soap. You can also use your spoon for that. I'm just kind of spooning some of the bubbles out of mine. So part of the reason why we added the salt is salt will help the DNA separate. DNA is polar, salt is polar. Those two molecules will work together. The soap helps break down the cell membranes to release the DNA. And the rubbing alcohol is going to help separate it, because alcohol and water do not have the same density. So the alcohol will form a layer on top of the water, which is what we want. And the DNA will be attracted to that alcohol. And so it's going to start to condense right where the water and the rubbing alcohol meet. All right, so now I have a glass that looks like this. I'm going to go ahead and get a paper towel. Now with my paper towel, I'm just folding it up like this. I'm going to use my paper towel to just get some of that soap out of the water and out of the way. All right, so now I have a nice clear boundary here that's not soapy. All right, it's kind of important that that boundary not be soapy. Then I'm just going to add my rubbing alcohol. Now what I'm going to do is carefully pour the rubbing alcohol so it forms a layer right on top of my salt water. All right, so I don't need a lot, but I do need a distinct layer So I'm just kind of gently pouring it down the side of the glass so that it starts to form its own layer on top of the salt water. And in a few minutes, that's where the DNA is going to form. So I'm looking for this kind of white, stringy substance. That's going to be our DNA. And we should see that within the next 5 minutes or so. So good luck. If it doesn't work out the first time, that's fine. Feel free to try it again. And hopefully improve some of those results.